You're listening to Fire Ecology Chats, a podcast series by the Association for Fire Ecology.
Hello, everyone. My name is Bob Keane. I am the editor-in-chief of the internationally renowned journal Fire Ecology. And today, I'd like to welcome you to Fire Ecology Chats and let you know we're going to be talking about a very, very exciting paper. The paper we're going to be talking about today is Mojave Desert microbial communities show high resistance and resilience over 3 years despite widespread plant mortality following the Dome Fire. We've got Sydney and Arik. Why don't you two introduce yourselves? Sydney, why don't you go first?
Okay, hi, I'm Sydney Glassman. I'm an associate professor in the Department of Microbiology and Plant Pathology at UC Riverside, and I am a mycologist. I primarily study fungi and the impacts of fire on soil microbial communities.
Hi, my name is Arik Joukhajian, and I'm a recent PhD graduate from Sydney Glassman's lab at UC Riverside in the Department of Microbiology.
Great. Well, let's hear about this paper, Sydney. Can you tell us a little about it?
Yeah, sure. In 2020, some of you may remember the front cover of the LA Times had 1 million trees killed in the Dome Fire, the charismatic Joshua trees. And it's been increasingly happening that there's these desert fires that are killing tons of Joshua trees. The Dome Fire was in 2020, and then in, I think it was 2022, there was the York Fire. So Joshua trees are getting burned. And I am someone who studies fires. I study the impacts of fires on soil microbes. So I went out and I set up some samples. And what I did was I followed the same study design. I had already been sampling fires in a chaparral shrubland nearby in Southern California. And so I set up the exact same experimental design where I had 6 burned plots and 3 unburned plots. And each plot had 4 subplots. And what I did was I sampled soils and we froze them so that we could do molecular sequencing. So the idea is that we were going to do molecular sequencing to identify the soil bacteria and the soil fungi from these communities. And I also partnered up with Lynn Sweet, who is a botanist in the Center for Conservation Biology at UC Riverside, and she helped me identify all of the plants and measure things like plant mortality at these sites. So we were looking at plant mortality and then relating that to the soil microbiomes. And one thing that was really cool when we got there is that almost every single tree was covered in these orange fungi. All over the branches and the bark was covered in orange fungi, Neurospora fungi, which are actually a model organism in molecular biology. Lots of Nobel Prizes have been won about Neurospora, and it was all over all of the bark.
That is amazing. People don't realize the importance of fungi in the environment and in the ecosystem, especially after fire. So, Arik, why did you do this particular study? What were the reasons behind it?
Yeah, so we were taking advantage of this opportunity because we don't see a ton of desert fires occurring in general, and we've seen a lot of consistency across what happens post-fire in other systems like forests and the drier chaparral environment in California, but we just don't often have this chance to study the impact of fire on desert microbes, so not a lot is known.
Desert fires are increasing in severity and extent across California, and we have no idea what the impacts of that's going to be on soil microbiomes. We know that Joshua trees are not adapted at all to these high severity fires. They basically don't recover at all, but we don't know anything about the soil microbiomes associated with them. For example, we knew that Joshua trees had arbuscular mycorrhizal fungi, which were symbiotic fungi associated with them, but until more other recent paper, no one had ever characterized arbuscular mycorrhizal fungi associated with the eastern Joshua tree. So his other concurrent research was about characterizing the arbuscular mycorrhizal fungi associated with the eastern Joshua tree. So we had no idea at this time, what are these mycorrhizal fungi? Are they dying? Are they going to help the trees recover? Is them dying or any of the other soil microbiomes that might be associated with these trees, the bacteria and fungi, having anything to do with recovery of these trees and the recovery of these landscapes? We know that deserts specifically rely on soil microbiomes a lot for things like carbon cycling and nitrogen cycling, and that's all driven by microbes. Are the microbes dying from this fire? Are they going to recover? All of that stuff was completely unknown.
That was the most interesting part of the paper is that you're actually breaking new ground here. It was incredibly interesting to read. So, Sydney, you think you could give us a little peek at what the major findings are from this study?
Yeah, so the first thing that we found was that there was massive death. The trees continued to die over 3 years. So what we found was that most of the trees had like some green leaves on them when we first sampled, but then over the course of the 3 years, we ended up with 86% mortality of the trees. And the most shocking thing was that even though like basically every tree died, the soil microbiomes had almost no impact. The richness was barely affected, and if it was affected, it increased after the fire. This was shocking. This was completely different than any of the other systems I've been working in. Joukhajian. I have done similar sampling in chaparral forests where you have 80% mortality of above ground plants and you have basically 80 to 90% mortality of the microbes. But in this case, the soil microbes were not that affected. And we thought, oh, maybe there's a lag effect. Maybe like the trees have died, but maybe we need more time. So that's why we kept sampling over 3 years. And it's still like even 3 years later, there weren't necessarily big lag effects. The soil microbiomes, the biomass and riches were barely affected. However, the composition was affected. Arik, do you want to talk about that?
Yeah, we saw some shifts in communities in between these burned and unburned communities over time. And some of the things driving bacteria and fungi to shift across these burned and unburned communities were actually not the same across different groups. For example, fungi increases in plant richness were driving shifts in the burned communities, while that had no effect on things like bacterial and archaeal communities. And meanwhile, those groups were being more affected by changes in soil nitrogen, things like ammonium and nitrate.
So after fires, one thing that we found, this has actually been known for over 100 years, that there's these things called pyrophilus microbes. These things are rare or absent pre-fire, but they massively increase in abundance after fire. And we found them after really severe wildfires in forests and in chaparral shrublands. I found them repeatedly in different studies I've done in different environments. What was surprising is that some of these taxa still showed up after the dome fire, even though there were like really minimal effects on microbial biomass, microbial richness, and like pretty small compositional effects, but there were still certain taxa like Neurospora that really increased in abundance after this fire. And then bacterial species too that are abundant after severe wildfires also showed up. So even though like the compositional differences overall weren't that big, still this fire did increase these pyrophilus microbes. And Arik is doing some really exciting follow-up work on looking at some more in-depth network analyses of these communities. So this is going to be a future study that he's going to be working on, looking at bacterial fungal interactions.
The fact that this work was done basically identically to work following a chaparral wildfire allows us to do a much better comparison of how the succession changes in the desert and how that might differ from a very wildfire adapted environment like the chaparral. And the fact that we have so much data on the microbes and at so many different time points allows us to do some really in-depth analysis looking at some of these microbes that we see that are pyrophilous, as well as dig deeper using network analysis methods to identify taxa that might not necessarily be the most abundant in the community, but might show up to be keystone species within the network plots.
I do want to say something. This is future work that we're working on publishing, but basically what we found is that these bacterial fungal interactions are really changing after the fires. So our current work that we just published is sort of pointing to this, that you have like not really big effects on biomass and richness, and the communities are really resistant to change. They're resilient, meaning that they're returning to their pre-fire communities and not changing very much over time in terms of like overall biomass and richness, but the community composition is having these slight changes such that they have these pyrophilus, fire-loving microbes, but also such that bacterial fungal interactions in particular are really shifting in these burn plots, driving probably whatever is happening after this fire in terms of soil stabilization or biogeochemical cycling.
One thing that our readers really, really love to hear is what are the management implications of these exciting findings? Sydney, do you want to take that one?
Yeah, so I had originally been talking with the park rangers of the Mojave National Preserve about this research. They did such a big study. They had a massive, massive effort where they had like, I think hundreds to maybe even thousands of volunteers trying to regrow these Joshua trees after the fire. And they were planting them and they were watering them, trying to get these things to grow. And I really thought like maybe one of the things that they need to do is to amend them with soil microbiomes, or maybe the mycorrhizal fungi might be important to help these plants grow. And originally what my plan was that we would do this analysis, see what's actually happening, how the fire is affecting the soil microbiomes, and then we were going to do a greenhouse study to test how a soil amendment might affect the recovery of these eastern Joshua trees after fire. But then when we found our results, where we found like the microbiomes are really not changed very much by the fire, we realized, okay, it doesn't make sense to do a greenhouse study. It doesn't make sense to do soil amendments because clearly those things aren't really affected. So I think that what we found is that the soil microbiomes are really, really resistant. Like even though the above ground biomass is not doing very well, like, I mean, these trees are not returning, 86% mortality, and not really a lot of recovery. There's a little bit of seedlings, but not that many. The soil microbiomes are persistent, so they're able to survive. And it actually seems like we don't necessarily have to do that much management intervention to get the microbial communities to recover.
Wow, that's amazing. Yeah, so kind of come to the end of this podcast. Arik, do you want to recognize any funding agencies or other co-authors?
We got a lot of help from Melanie Davis in the field, as well as Lynn Sweet, statistical help from my former lab mate, Fabi Polito-Barriga.
Fabi Polito-Barriga actually helped me set up the plots because I set them up before Arik joined my lab. So she was the one who came out and helped me go to the desert and set everything up while everything was totally burned, and we found the plots. And then Lynn and Melanie met us in the field and were helping us with all the plants. We definitely could not have done the plant work without Lynn and Melanie. They were extremely critical. And we really want to thank Deborah Houston and Drew Kaiser and all of the people at the Mojave National Preserve for allowing us to sample, and also the Sweeney Granite Mountains Desert Research Center at UC Riverside. They allowed us to stay and support us in our research, and most of this research was actually supported by UC Riverside and my startup. We also had funding from the Shipley Skinner Conservation Science Award that was to Arik, and then also I got support from the United States Department of Agriculture what does NEFA stand for? The USDA NEFA Agricultural Microbiomes Award also supported this research.
Well, I want to thank you two for taking the time and telling us about this really exciting study. I have so many more questions. And you know what I'm going to do? I'm going to go back and reread that paper because it's a really exciting paper. And I would suggest you listeners go ahead and do it too, because you know what? You can download them for free because these two people have already paid the freight for you to download it. So you can download it and learn all about soil biota and its relationship to fire. Again, thank you, Sydney and Arik, for your time. And everyone, please tune into the next Fire Ecology Chats.
Thank you for listening to Fire Ecology Chats, a podcast series by the Association for Fire Ecology. If you enjoyed this episode, subscribe to our show and leave us a review. To learn more about fire ecology and support our future work by becoming a member, visit fireecology.org. fireecology.org.